Thank you, Scott. You clearly won the toying toss. Anyway, we now have Dr. David Gandara to update us on the best of SWOG in lung cancer.
Thanks, Chuck. It's a great pleasure for me to present on behalf of my leadership colleagues in the Lung Committee and the Lung Committee at large. I've entitled this presentation, Innovation Meets the NCTN, or National Clinical Trials Network, because I think this is quite a transition, and when the federal government, assuming the federal government reopens, in a few months we'll all be in this brave new world of the NCTN. Now, I can tell you someone in a very high place suggested I might entitle this presentation Innovation versus the NCTN. But I think that's actually wrong thinking because although there are considerable challenges here, as the old saying goes, in every challenge there are opportunities. So this presentation will address how the Lung Committee views the challenges and also how we might take advantage of the opportunities. So this is my own slide about innovation meets the NCTN. And for SWOG at large and the Lung Committee in particular, the issue is how would we be successful? How do we address the challenges? How do we take advantage of the opportunities? And as the Lung Committee members know, I have proposed that we follow the 4 T's, or the 4 trans: transition, transdisciplinary, translational, transformative. So the transition is where the greatest challenges lie, in my opinion. There are steering committees. In our case, it's the TMSC, Thoracic Malignancy Steering Committee. And increasingly, they have been mandated to not only review and approve proposals from the groups, but also to propose and design the trials themselves and to assign the groups to carry out those trials. That's a challenge. In addition, unbeknownst to myself or any of the other Lung Committee chairs for the other groups, the NCI formed a thoracic malignancy working group to determine future priorities on a national basis for research in lung cancer without any input from the groups. And we've been presented the priorities, and already I have heard twice that proposals don't meet the priorities of this group, which contained and was chaired by many people that are not even in the lung cancer field. These are the challenges. Well, what are the opportunities? Transdisciplinary in the past usually meant multimodality. Increasingly, transdisciplinary means that we must take advantage of the resources either within SWOG or external to SWOG in disciplines such as genomics, epigenomics, preclinical modeling, and imaging, just for some examples. To be successful, our studies need to be translational. Everybody understands what this term means. Well, what do I mean by transformative? Here I mean that in order to be successful, our new Lung Committee trials need to either have the potential to change our understanding of the biologic concepts of the disease or patient subset under study, to change clinical practice, to change the standard of care. And what I'll do in this presentation briefly is to talk about 2 developing SWOG trials, S1400 and S1403, which I think address the 4 trans. So the first of these is the master protocol, as it's been named, for squamous cell lung cancer, S1400. This is chaired by Dr. Vali Papa, as I call her, from MD Anderson. This is already a 1.5-year process. It is The term unique is overused. This is absolutely unique. This is a public-private partnership which the groups have never seen before. And I would like to offer my personal thanks to Dr. Larry Baker, who shepherded this proposal during its early gestation. Okay, and I use this slide to introduce the topic because for those of you not in lung cancer, you hear the term non-small cell lung cancer. This is from a paper published earlier this year in the JCO about genotyping from Tina Lee and our group at UC Davis. And it shows the evolution over the last few years from non-small cell lung cancer considered as a single disease to histologic subtypes to now a multitude, both in adenocarcinoma and squamous cell cancer, of genomic subsets, some of which are sizable, some of which are rare by anyone's definition. A rare disease, Anne, within lung cancer. Well, what are the unmet needs in clinical trials now that we think about this disease as a multitude of genomic subsets? How do you develop drugs for uncommon or rare subtypes? And I can tell you that there are companies, for example, that have trials for BRAF-mutated non-small cell lung cancer. How do you go to a patient, or how do you go to an investigator and say, screen your patients for this entity, which represents a fraction of 1% of the population? How do you apply new technologies such as next-generation sequencing in the presence of clinical trials in order to screen patients for these genotypes? How do you, in a symptomatic patient with advanced stage disease, have an acceptable turnaround time for something like next-generation sequencing so that within 2 weeks you can assign a patient to a therapy? And lastly, how do you energize the FDA approval process for new drug and biomarker combinations, companion diagnostics? And so the master protocol is designed to address some of these goals. This had its gestation at a meeting in February 2012. Again, I think a unique event. It was a joint meeting between the FDA and the Thoracic Malignancy Steering Committee. We also had representatives from pharma, about 10 companies. We had representatives from the EMA and several other organizations. And one of the outcomes here, because I just have a short amount of time here, was an action item to develop a master protocol, and SWOG rose to this challenge. So this shows the, some of the leadership for this protocol: Master Lung 1, S1400, a biomarker-driven, multi-arm, phase 2-3 registration protocol in squamous cell lung cancer second line. And you can see it has the entire North American intergroup involved, as well as a number of SWOG leaders playing pivotal roles in this trial. But as I'll show you in a minute, the governance and the participation in this extends way beyond the NCI and SWOG. Shown here is the rationale. I don't really have time to go through it in detail. These slides will be on the SWOG website. But the theme here is to have a homogeneous patient population and consistent eligibility in a multi-arm trial, with each arm opening and closing and functioning independent of the other arms, but having an infrastructure which is self-sustaining, so that as arms close, either due to failure or success, new arms can replace them. To transform our drug development process so that we look for large effects, as was mentioned by Craig in his presentation. To facilitate screening large numbers of patients, we anticipate about 1,000 per year in this protocol, for multiple targets, which would for the first time be presented to the FDA, and actually with an FDA in partnership on the trial, as a registration platform. Bring drugs that are safe and effective to patients sooner, approve drugs faster. This shows the design for the protocol. I realize this is complex, But think of these as multiple arms, each of which is biomarker driven, so biomarker A, biomarker B, biomarker C, etc. Each one of these having a targeted therapy selected through a drug selection committee with input from multiple entities. This has been going on for over a year. 27 drugs evaluated, 16 pharmaceutical companies engaged in the process. So you can see for biomarker A, targeted therapy A, there would be the drug versus a standard chemotherapy. The same in B. For C, that drug marries well with chemotherapy. So it's drug plus chemotherapy versus chemotherapy alone. And you can see in biomarker D, this is an EGFR-related drug, so erlotinib is the partner and also the comparator. And you can see also a non-match arm. So this might be a drug or a category of drugs where the biomarker is not yet totally clarified. There are 2 or 3 possibilities, and the FDA is going to work with us with bridging studies to ensure that this also could eventually lead to a registration. Phase 2, 3, so with interim endpoints if there's failure. Now, here, no biomarkers, no drugs. Here, biomarkers, drugs. So we have selected the initial 6 drugs, but as the old saying goes, if I told you today I would have to kill you because there are large dollars at stake here. But you can see one of these drugs will be a PI3 kinase inhibitor, another against CDK4/6, FGFR, and HGF, and you can see the marriage there, and you can see the proposed biomarkers that would be tested. Oh, and then I should go back and say the unmatch will be a PD-L1 antagonist. And this shows how a patient would enter the trial, how they would be screened. The screening process will be the next generation sequencing platform by Foundation Medicine. They are working with us and the FDA to assure that at the end of the day this can be a registration platform. This will be the first time that next generation sequencing has ever been put in this position. So that you can see then a patient could be randomized to either investigational arm or standard of care. And you can see also on the left here that in this case, patients with advanced stage squamous cell lung cancer getting first-line platinum-based therapy, their tumor could be submitted and pre-screened so that they are ready to be enrolled at the time of failure to the first-line therapy. And so that will allow rapid accrual and decrease the issue about symptomatic patients waiting for results. You can see the organizers include the Friends of Cancer Research with Ellen Siegel, a very important aspect of this, the NCI Thoracic Malignancy Steering Committees, the FDA, the foundation of the NIH, where the funds will go from pharma. Participants, entire North American intergroup, up to 1,000 patients per year to be screened. This trial, we hope, will always have 6 arms open simultaneously. Obviously, the prevalence rates of these genotypes varies considerably. There will be a randomization if a patient's tumor matches twice, for example, so that we assure that the rare genotypes arms also are completed in a timely fashion. And we anticipate always having at least a 60% hit rate, but as you saw, we have the non-match arm to account for the non-hits. This shows the governance structure, which is considerable, and you can see it includes the NCI, the FDA, the Friends of Cancer Research, the Southwest Oncology Group, and the foundation of the NIH. So this protocol is designed to change the way we do business. And for those of you that are involved in this protocol, you probably know that I have made this statement 100 times to every one of these stakeholders, because people tend to retreat into what they have done and what they know best, and that is what they have done for the last 20 years. So we intend to change that if we're successful in this protocol. And I can tell you that around the world, people are watching this. Also, colorectal cancer wants to be the next master protocol. They already have this blueprint. So first, we need to be successful, and that will also depend on the participation of all of you in the audience. Okay, the second trial I will just briefly close with is SWOG 1403, Afatinib plus or minus Cetuximab, an EGFR-mutated non-small cell lung cancer. This is led by Tom Lynch, Sarah Goldberg, in the audience, our Young Investigator Awardee, and Katie Politi for Translational Science. So this is a very important concept in oncogene-driven cancers. And non-small cell lung cancer, of course, is a very good example of this. This schema for a clinical trial design paradigm was presented by Mary Redmond and myself at an ASCO educational session this year. And we talked about how you might assess or how you might circumvent acquired resistance by upfront therapy. And so we talked about one arm of a trial being a first-generation agent, and if we talk about EGFR mutation, this would be a drug like erlotinib. The second arm being a second-generation drug, this would be afatinib, and then a multi-drug targeted therapy, and you can think of several combinations, but the one we're looking at here is afatinib plus cetuximab. And the S1403 trial will look at these two last arms. So this shows the design. Again, this is in first-line therapy, treatment-naive, advanced-stage non-small cell lung cancer, EGFR-mutated. The steering committee, and I think, again, this is taking an opportunity, we said if we're going to study this paradigm, we should study it in first line, because that will be the potential greatest transformative component of this trial. That's our chance to have the greatest impact on survival, and they agreed with us. These are the data from our colleagues at ECOG, William Powell and his colleagues, also Katie Politi, of the trial that they did in EGFR-TKI refractory patients, so patients who had failed erlotinib. And they were treated with afatinib plus cetuximab, and you can see the response rate was about 30%, but you can see disease control rate and the waterfall plot are very impressive. Now, the preclinical modeling here, and this is why we need better preclinical models, this was in a GEM model, suggested that this drug combination would only be effective in refractory patients with the resistance gene T790M. And in fact, as you can see, that's in red. The green is the patient's tumors that did not have the resistance gene, and it was equally effective. So part of the rationale for our study is also to find out why this combination is T790M independent, what's the mechanism of action. Afatinib alone, I should say, in the refractory setting only has a response rate of about 10%. So why is this different? And the combination doesn't work with erlotinib. So again, there's something about the second-generation agent which is important. And lastly, you can see in red here, and again, this will be a first time. As part of this protocol, we propose to have patient tumor biopsy at the time of progressive disease, and approximately 1/3 of the patients on both arms of the trial, and that in addition to a wide genomic analysis of those tumors, that there would be implantation into patient-derived xenografts to develop a national resource for the study of acquired resistance to EGFR-mutated cancers. And this will engage our SWOG Translational Science Center. This will be the initial pilot project, hopefully for that group, which, as you know, in addition to SWOG, includes our basic science cancer center collaborators, the Jackson Laboratory, with the PDX models, and Cold Spring Harbor with their expertise. And I'll just finish by showing a slide that I've shown in this forum before. This is the initial pilot studies from the UC Davis and Jackson Laboratory at JAX West in Sacramento, where we've established this array of genomically characterized PDXs for study. What I will tell you, though, is that all of these come from different situations. The PDX resource we'll be developing for 1403 will be a homogeneous group of patients, all treated the same way, with a control group. There will be nothing else like it. So transition, transdisciplinary, translational, transformative. The SWOG Lung Committee seeks to address the challenges and to take advantage of the opportunities. Thank you.
Great. So we do have a few minutes for questions for Dr. Gandara. Really is a brave new world.
David, that was a great talk. At the breast meeting yesterday, I can't remember, Deborah Tripathy presented the MATCH study. How will the lung cancer study interact with the MATCH? Maybe you could tell what the MATCH study is, but how will you be outside that, or is this true and unrelated? Or
so, as Dr. Baker knows, I introduced this presentation with challenges versus opportunities. So we do have a number of members from the investigational drug branch of the NCI on our drug selection committee, and on the first one of these, the head of that group said, you know, maybe we should rethink this entire master thing. This is after a year of working on this. He said, you know, we're going to have this NCI match trial. Maybe you don't even have to do this master. Maybe we can just put the patients on the match. So the match is, in my mind, exploratory. And it seeks in multiple tumor types, whereas there is a common genotype to treat the patients the same way and gain some information. The master protocol is a registration strategy in every possible way. The FDA is working with us to develop the case report form so they will be registration ready. The companion diagnostics from CEDAR. So it's a very, very different sort of thing. And I think once we explain that to the NCI, they have bought into it completely. So,
are they mutually exclusive in terms of the same patient?
yes, they're mutually exclusive. Okay, thank you.
Thanks, David.